All right, thank you very much, Andy. So I'm going to switch gears a little bit here and tell you about some of the basic research done in Parkinson's. And as Dr. Espe alluded to, we need to understand some of the basic molecular mechanisms of the disease. Again, no one Parkinson patient is the same. The disease will not progress the same for everyone, sort of like they're all different cancers, but there are basic underlying mechanisms and pathways that are common along in both cancer and with Parkinson's, and a lot of the basic research, is trying to delve into that. And I want to highlight just a couple main topics that you might not have heard yet, but some people have been asking about. Something called the microbiome, having to do the gut and Parkinson's, and also the protein that is corrupted and misfolds in Parkinson's and is thought to underlie perhaps Parkinson's or not, there's a debate, alpha-synuclein. So let me just start here with the gut-to-brain model of Parkinson's disease. Based on work in the pathology of human brains, it's thought that Parkinson might start in the gut, right? And then somehow be transferred up to the brainstem and then to the front of the brain, and then finally, okay, there's no echo now, good. Finally, where it gets to the middle of the brain, where the dopamine cells are, the ones that degenerate. So how does this do this? Can you show that it can do this? So what this group did, I think they were at Johns Hopkins, was that they took toxic alpha-synuclein seeds of this bad protein, and then injected it into the gut, certain layers of the gut. What they then found that it's then transmitted, and you can see in that diagram on the right there. So you can see the injection needle that's in blue. They inject it into the gut. This toxic seeds of alpha-synuclein then encountered normal, already present or endogenous alpha-synuclein, corrupted it, right? Turned it into abnormal form, and then it was transported sort of like a pneumatic system or something back up the brain through those two nerves that you see there. That's the vagus nerve to the back of the brain. It encountered perfectly normal alpha-synuclein there, corrupted it, and then was relayed on to another neuron. That's sort of a relay, a nerve-to-nerve cell relay, up to finally it encountered the dopamine cells in the midbrain, and that caused some clumping and the cells to degenerate. It also caused, injecting this toxic alpha-synuclein into the gut, also caused motor symptoms, so gait problems with the animal. You can see the animal shaking there. And non-motor symptoms. So this was good because previous models could only maybe model just motor problems, but now they found cognitive problems too with the animals when they were asked to do certain mazes, for example, right? But one of the interesting things here is that this Parkinson's pathology and these symptoms require normal alpha-synuclein already present in the cells. And I'll show you why that is on the next slide. So these are just three diagrams here, and on the left is what I just showed you. So if you inject these toxic seeds, it corrupts alpha-synuclein, it goes up to the brain through these nerves, and you get this spreading of toxicity, of pathology, and the motor and non-motor problems. However, in the middle panel, that's labeled vagotomy, that simply means cutting of the vagus nerve. If you cut that nerve and inject those toxic seeds in the gut, then very little happens, right? So that's because then this toxic form cannot get back up to the brain, all right? And there's some evidence in humans with also vagotomies that's both supports and doesn't support this theory. And interestingly, finally in the right diagram, you inject those toxic seeds, but in a mouse, because we use animals, in a mouse that has been engineered that it doesn't have any normal endogenous alpha-synuclein, right? So there's nothing there to corrupt. When you do that, you don't get any Parkinson's-like symptoms or any pathology, right? So that's good evidence that supports this theory that Parkinson's can start in the gut. It doesn't mean it does, but it certainly says it's possible. And it's a working theory of many, many groups in Parkinson's basic research. Okay, continuing along the gut theme, gut bacteria. You might have heard a lot about the terms gut microbes, gut bacteria or the microbiome, right? So that's basically bacteria, millions of bacteria that populate human gut, animal guts, for example. What was shown by this group was that the gut microbes, so these gut bacteria, promote alpha-synuclein-mediated motor deficits and brain pathology, right? So, I'll show you a diagram on the next slide. What that picture is there is of a immune cell in the brain, so-called microglia. So, and if you deplete gut bacteria, you can do that in animals. So take away all the bacteria in a mouse gut. It reduces brain inflammation. Brain inflammation is associated with progression of Parkinson's disease. And it's thought to be caused by something called short-chain fatty acids. And what are those? Those are basically, it's kind of a lipid, but it's a breakdown product of bacteria, right? So it's simply a byproduct of bacteria. But these molecules can influence immune cells, and they also worsen Parkinson's pathophysiology. And interestingly, human gut microbes taken from Parkinson's patients and injected into the gut of animals induce worse motor deficits in mice than normal. And again, I'll show you this on the next diagram. And it's not really that complicated. It's just, if you look on the left, it's what I just described. There's the typical, it's called microbiota, typical gut bacteria in an animal model. It releases into the bloodstream these short-chain fatty acids. They influence the immune cells, they activate them, it's a bad thing, it gets to the brain, it causes Parkinson's-like symptoms and pathology. Now, if you have an animal that you take away the bacteria in the gut, like in the middle, what happens? There's no short-chain fatty acids, so there's no signaling, you don't stimulate the bad glia, the bad immune cells, and therefore you get little to no pathophysiology in the brain. Interestingly, however, if you have this mouse, and in its gut you inject bacteria derived from a Parkinson patient, you then get enhanced motor dysfunction in the brain. So there's something about the bacteria, it's thought, in Parkinson's patients that is detrimental, to the brain. So the bottom line, again, is that signals from these gut microbiomes regulate motor problems, pathology, degeneration in the brain. And again, those two, the alpha-synuclein and the microbiome are really hot topics in Parkinson's, not only Parkinson's, but in many fields of neurodegeneration and neuropsychiatric diseases. Switch gears a little bit here to another topic, and I thought this was very interesting. A group has found that a new molecule stops damage to nerve cells. And they named it, cleverly or not, synuclein D. Right? It's a play on the word alpha-synuclein, of course, but it inhibits the clumping of alpha-synuclein, and it's this clumping, this misfolding, that is thought to really detrimentally affect brain cells. So what they did is they screened 14,000 molecules to identify the new molecule. They did this mostly in test tubes and in culture dishes. And what they found also, so it reduces the clumping. You can see on the upper left, so without this new molecule, you can see like the big clumps and these fibers around, but treated with, you can see it almost eliminated all of the clumping. So again, this was done in vitro or in test tubes, for example, but they also did it in an animal model. They injected it. And they found that it rescues dopamine cells from this alpha-synuclein clumps that induced cell death in these dopamine cells. So the bottom line is that, and this is at the very beginning stages, but the bottom line is that this molecule, synuclein D, may have therapeutic applications in the future. And this group is going on to test it into more advanced animal models. And then finally, in the last just example of what I'd like to tell you about, again, I'll get back to alpha-synuclein, but it's another function that really hasn't been discussed or found about alpha-synuclein before. That's interesting. I didn't touch anything. Anyways, so the normal function of alpha-synuclein is thought to be at the ends of nerve cells. So at the ends of nerve cells, in cell-to-cell communication, anyways, chemical messengers are released from the end of a nerve cell onto the next. And alpha-synuclein is thought to kind of coordinate that, kind of improve the scaffolding and kind of marshal the right chemical messengers to the edge and then get them outside of the cell so they interact with the next cell and then communicate. But now this other group has come up with maybe a new function of alpha-synuclein that's in the nucleus of the cell, right? So you have a cell. Is this? Nope. So, if you have a cell body, it'd be nicer with a picture, but what they found is that alpha-synuclein actually enters the nucleus of the cell also. Not just the end of the cell, but the nucleus, right? And it's involved in DNA repair. So most cells, you have DNA, there are so-called double-stranded breaks, but there's always a continuous kind of degeneration or injury to DNA, but that the cell has its own repair mechanisms that kind of that fix it up. And it's thought that alpha-synuclein participates in this. There we go. So alpha-synuclein regulates DNA repair in the nucleus. That red, you can see the red blotch over there, that's the nucleus of the cell. So it's a new function for alpha-synuclein. It helps in DNA repair. So, but what this group also showed that you can see a Lewy body there. You've heard Dr. Espay talk about Lewy bodies. That's where the alpha-synuclein clump is misfolded and it's kind of packed into these Lewy bodies. And what it's thought is that alpha-synuclein is somehow sucked out of the nucleus, it's corrupted and gets put into these Lewy bodies, and therefore there's not enough alpha-synuclein left in the nucleus to do DNA repair. So the bottom line is this is a new twist on the function of alpha-synuclein, right? So there's unrepaired DNA damage because you don't have enough alpha-synuclein in the nucleus because it's out there in the Lewy bodies, and that this may trigger cell death. So just to summarize now, again, I've told you, and I hope I've convinced you a little bit, that there's good evidence that Parkinson's may start in the gut and spread up to the brain, from the back to the front of the brain. The gut bacteria regulate Parkinson's, the pathology in the brain, the motor deficits in the brain, the microbiome. There's a new molecule, synuclein D, that prevents this clumping and cell degeneration. And then finally, there's a new function, DNA repair, for alpha-synuclein. And with that, I guess I'll end for now. And we'll take a short break, and we're going to set up the table for the panel session. So if anyone needs to go to the bathroom or that, but we encourage you to stay around if you can. And then we'll answer as many questions as we can in the next panel Q&A group. So thank you very much for your attention.